And purine, we are coming into the chapter proper. Purine nucleotide synthesis. How does this nucleotide synthesis happen? It happens in three ways. The most important way is the de novo synthesis of purine from the amphibolic intermediates. Yes, from its amphibolic intermediates. So it'll add nitrogen, it'll add carbon from each of from any amino acid or from tetrahydrofolate or any methyl donor. Yes, and it will form the ring structure, then on top of a ribose 5-phosphate. So this is when a step-by-step addition of nitrogen carbon atom happens, then that is going to be the de novo synthesis. That is from the amphibolic intermediates you're synthesizing. Now other two ways are the salvage pathway. So after degradation of your nucleic acids, those nucleic acids, whichever the broken product, those broken products will get utilized. So in which case with the free purine, if you are going to add a sugar and a phosphate, that is nothing but phosphoribosylation of purines. Now to a purine nucleoside which has a purine and a sugar to which you are phosphorylating it, then that is going to be phosphorylation of purine nucleosides. These are the two important ways by which salvage synthesis happens, but most important way, abundant way is going to be the de novo synthesis of purine, right? So de novo synthesis of purine will give us this purine ring. So this purine ring, the important, very important thing is going to be the sources of purine ring. Yes, it is going to be the sources of purine ring. Now let's learn this. We have already learnt about the numbering of this purine ring. It has two rings. One is a 6-membered ring, the other one is a 5-membered ring, right? So from the 6-membered ring only you have to start numbering. So this is N1, yes, and 2, 3, and you have 4, 5, 6 and N7 and 8 and N9. Okay, so to this N9, we know the sugar will bind to this N9 of purine, right? And what is the source of N1? The source of N1 is going to be aspartate. Yes, first alphabet A. So N1 source is going to be aspartate. So please memorize this now itself. So while learning the purine synthesis pathway, it is going to be very easy, right? So N1 is going to be from aspartate. And if you see C2, yes, C2 and C4 are going to be C2 and C4, it will have, sorry. C2 and C4, C2 and C8 will have tetrahydrofolate acid source. Okay, so two carbons, no? 2 and 8. So 2 into 4 is 8. So tetrahydrofolate has 4 tetra in it, right? So just remember it that way. So C2 is from N10 formyl tetrahydrofolate and C8 from N5 N10 methylene tetrahydrofolate. This is a source from Harper. Recent edition Harper also has this. So please memorize this. Okay. And glycine. If you take glycine just has, what is the structure of glycine? It has a carboxyl group to which the carbon is attached and an amino group is attached to that carbon, and the side chain is nothing but H2. Yes. So entire structure of glycine is being incorporated into this purine ring. That's why it's a donor of C4, C5 and N7. Okay, glycine is a donor of C4, C5 and N7. So entire structure of glycine is inside this purine ring, right? And bicarbonate or the respiratory carbon dioxide is a source of C6. Okay, it is a source of C6. And last but not, N3, N9, the source of which is going to be glutamine. Alright, so what are the different sources? One is aspartate, yes, and the other one is the tetrahydrofolate, glutamine, glycine and carbon dioxide. So the carbon donors will be tetrahydrofolate and carbon dioxide. The nitrogen donors will be, the amino acids as donors will be aspartate, glycine and glutamine. C4, C5 is also given by glycine. Okay, so this is the sources of purine ring. So de novo purine synthesis. So dietary nucleic acid, the breakdown of dietary nucleic acid is not going to be used in the de novo purine synthesis. Okay, but it's not going to be used in the formation of purine nucleotides. But de novo purine synthesis abundantly takes place in the liver. Okay, abundantly takes place in the liver. Whereas in brain, RBCs, WBCs, the enzymes which are involved in this de novo purine synthesis are relatively in lesser amount, or no enzymes are present there. So that those areas like brain and RBCs will use salvage pathway, right? But de novo purine synthesis will happen in liver. Okay. Now, purine ring is nothing but purine ring. How is it synthesized? We have a ribose 5-phosphate. So we have a ribose 5-phosphate, on top of which we are going to start adding the nitrogen and carbon, and we are going to make the purine ring. Okay. So this is a preformed ribose 5-phosphate, on top of which we are going to add the nitrogen and carbon by a series of reactions, and we are going to form the purine nucleotide. Okay. Now, yes, so where does the ribose 5-phosphate come from? Of course, it comes from the HMP shunt pathway. Alright. So all these enzymes are present in the cytoplasm of a cell. Okay. Now, first step. So first step, we can also call it as the preparatory step. Yes, we can also call it as a preparatory step. So this preparatory step is nothing but the formation of this PRPP. Okay, PRPP is very important here. After this PRPP, it will enter into either pyrimidine synthesis also or purine synthesis. But here the first step, preparatory step, is going to be the ribose 5. Already told you, ribose 5-phosphate is only going to is the substrate here. So the ribose 5-phosphate, if you see, yes, it has this ribose to which a phosphate is attached. Yes. And the enzyme is PRPP synthase, and it uses an ATP. Why does it use an ATP? The 2 phosphates of this ATP will be incorporated to this ribose 5-phosphate, forming phosphoribosyl pyrophosphate. Yes, which is nothing but our PRPP. Yes, which is nothing but our PRPP. Now, what are the inhibitors of this enzyme? The inhibitors, of course, the inhibitors of this enzyme will be the products of this pathway. Yes, the purine nucleotides. Whereas activator of this enzyme is going to be phosphates. Okay, ATP. Yes. And if you see, let's talk about each and every step. It is going to be very easy. Yes, even though they say in detail knowledge of the steps in purine synthesis is not essential for your exam purpose, but the overall understanding is going to be essential. So let us dive into it, right? So the first step, as we have already seen, we have the ribose 5-phosphate. To the ribose, a phosphate is present. To the OH group of this ribose, it is in the first position, the OH group, to which we are going to add 2 phosphates. Yes, and that structure is called as the phosphoribosyl pyrophosphate, and the enzyme is PRPP synthase. It uses magnesium. Of course, ATP will be utilized here, right? And what is the second step? This is going to be the committed step. Yes, second step is going to be the committed step here. Okay, and what happens in the second step? We are going to add the glutamine. Yes, so for remembrance sake, I'm telling you, first, on top of ribose 5-phosphate, the 5-membered ring is going to start first. Yes, start assembling first. After which the 6-membered ring will will start assembling. Okay, so this glutamine in the 5-membered ring, which nitrogen is being donated by this glutamine, we know it is N9. No. Yes. Yes, so that is why the glutamine donates its amino group. Yes, the glutamine donates its amino group, and as you can see, on top of this is the phosphoribosyl pyrophosphate. Now to this nitrogen is an amino group from the glutamine, which is obtained from glutamine, has been attached here. Right? So how does this happen? The pyrophosphate is being removed, and this alpha configuration is going to convert into beta configuration. And in this beta configuration, please look into this picture. Yes, the alpha configuration here, which is present here, is getting converted to beta configuration, and the amino group is being attached. Right? So this is catalyzed by the enzyme PRPP glutamyl aminotransferase. It is a very important enzyme. It plays an important role in the regulation of this purine nucleotides, right? So this first thing is we are adding the glutamine. The amino group from the glutamine has been attached to the PRPP. Yes, on removal of this pyrophosphate, this amino group is getting attached. So again, how will be denoted? On ribose 5-phosphate, the amino group is attached. Yes. So the next enzyme that is being attached, the next amino acid in the next step, third step, what happens? The next amino acid, which is the glycine, which is going to get incorporated. Now glycine, as you look into the structure, how is it? It has the carboxyl group. Yes, carboxyl group. It has a carbon, the side chain has hydrogen, and to which an amino group is attached. This is the simplest amino acid. We all know, right? It's the simplest amino acid. It is optically inactive. Yes, so that's why it's different from all other amino acids which are all optically active, right? So this entire structure of this is going to get incorporated into the purine ring. So in which case, what it will be a donor of C4, C5 and N7. Yes, C4, C5 and N7. So look at how it is being attached here. Yes, this is the product that we have obtained. How is it attached here? Yes, so here is the this is the ribose, this is the phosphate, so ribose 5-phosphate, on top of which you have added the amino group, yes, from the glutamine, and to this amino group, what is being attached? To this amino group, you are going to get this glycine. Glycine is going to get attached. Once again. Right. So is it visible now? Yes. So where did we stop? We have incorporated the glycine in our structure, right? And now look into this structure. This after incorporating the nitrogen, amino group from glutamine and the glycine structures. Now we are going to add N5N10 methynyl tetrahydrofolate. N5N10 methynyl tetrahydrofolate, that is going to be the donor of another carbon. Yes, it is going to be the donor of another carbon. So so what is that carbon going to be? It is C8. Okay, so it gives off its formyl group. So this is formyl transferase. So it gives off its formyl group. So how this is attached? So look at how it is attached. Yes, you have a ribose 5-phosphate, and to the ribose 5-phosphate N9 from glutamine is attached. To this N9 from glutamine, we have even attached the glycine. Yes, and to this glycine we have again attached the formyl group from the tetrahydrofolate. Is it fine? Yes. So from this, the next step is going to be addition of another glutamine. So we have two nitrogens which will be donated by glutamine, right? So glutamine will again give off its nitrogen group, and where does it get attached? It gets attached at this position. Yes, from C4 it gets attached. The amino group gets attached. Now we are going to close this ring. Yes, now we are going to close this ring. So the ring closure takes place. So here, look at this. We have the ribose 5-phosphate. We have the N from the glutamine. Yes, first glutamine, and also we have the glycine structures, 2 carbon and 1 nitrogen, and the carbon which is donated by the formyl group, for tetrahydrofolate group. Yes, and now the ring closure has happened. The second glutamine donates its nitrogen amino group that is being attached here. Yes, and that is nothing but our N3. Yes, fine till here. Yes. So anywhere you have doubts, if you want me to repeat, please do it in the chat box. Reply it in the chat box. Okay. Now, what is going to be the next step after we have closed this ring? So first we have synthesized which ring? The 5-membered ring. Yes. So on top of ribose 5-phosphate, we have synthesized the 5-membered ring, and then we also have this N3 from glutamine attached. Now we are going to add carbon dioxide. Yes, carbon dioxide is going to get attached. Now look at where this carbon dioxide is getting attached. So carbon dioxide is here. So it is a donor of C6, is it not? So you have to, the carbon dioxide must attach in this place. Yes, now after attachment of carbon dioxide, this is a carboxylation reaction, but this carboxylase does not need ATP or biotin, as that is the case in any of the other carboxylase enzymes, is it not? So that is one difference here. And now this after the incorporation of this carbon dioxide, the next step we are going to add aspartate. Yes, we are going to add aspartate. Look at the structure of aspartate. We have carbon, yes, attached to carboxyl group and amino group, and the side chain is going to be CH2COOH, right? And this is going to get incorporated here. It is aspartate is only going to donate its nitrogen. Yes, it is only going to donate its amino group. So to the place where, to the C6 place, this nitrogen is being attached. So this we have to remove. The side chain of the aspartate apart from the amino group has to be removed, is it not? So that removal is done in the next step. So this is nothing but the fumarate, that's the side chain apart from the amino group of the aspartate. So now that is being removed again in the next step. And the final step is, the next step, what is going to happen? N10 formyl tetrahydrofolate. We are left with only giving one carbon, is it not? So this N10 formyl tetrahydrofolate gives off its carbon. So again, the formyl group is being given here. So the formyl group attaches to the N3. Okay. Now, again, what is left of here? We have to close this ring, is it not? We have to close this ring. So ring closure happens. The ring closure happens in such a way that the 6-membered ring is now fully formed, right? So this is the ribose 5-phosphate, on top of which we have synthesized this purine, and that is nothing but inosine monophosphate. Okay, so the first form purine nucleotide is going to be inosine monophosphate. That is very, very important. The first form nucleotide is going to be inosine monophosphate by the de novo purine synthesis. Is it clear now? Yes. So what is so let's see the overview. Yes, I'll just tell you only the sources. So in the first step is nothing but phosphoribosyl pyrophosphate synthase. So that's the preparatory step. So starting from PRPP, we are going to start a purine synthesis. So purine synthesis, the committed step is going to be PRPP, glutamyl amidotransferase, because PRPP as such can enter into pyrimidine synthesis or histidine synthesis, like that. So here PRPP, glutamyl amidotransferase, is going to be the committed step of this de novo purine synthesis. So first I'll add a glutamine, then I'll add a glycine, then I'll add a N5N10 methynyl tetrahydrofolate, then I will add another glutamine, then a ring closure happens, then I'll add a carbon dioxide, after which aspartate is being added, then the side chain of aspartate is removed, then N10 formyl tetrahydrofolate again I will add, then on ring closure I get the inosine monophosphate. Is it clear, all of you? So in this way, if you learn this purine synthesis, it will be well and good, right? Yes. So in the next is, we'll just see, this is the committed step I told you, is it not? So this activator of this step will be the phosphoribosyl pyrophosphate. Yes, so which step is it? Glutamine PRPP amidotransferase. That step only, the first step in which we are going to give one glutamine, first glutamine. That is what I'm telling here. So the inhibitors will be the products of this pathway. So product of the pathway, till what we have seen inosine monophosphate. From inosine monophosphate, AMP, adenosine monophosphate, and guanosine monophosphate will be formed, right? So those will be the final products of this purine synthesis. So if there is more of products, of course, it is going to feedback inhibit the de novo purine synthesis. So in which case this committed step is going to be regulated by these inhibitors and activators, right? Yes. So next we'll see how from inosine monophosphate we get adenosine monophosphate and guanosine monophosphate. Okay, that happens in two ways. One way is that inosine monophosphate, yes, inosine to this inosine monophosphate, if you add a yes, so there is there is this adenosyl succinate synthase, right? So that from which the amino group is being attached to this nucleotide, then we get a adenosine monophosphate. Yes, then we get a adenosine monophosphate. So that is going to be yes. Yes, so that is going to be the synthesis of adenosine monophosphate, in which the enzymes adenine succinate synthase and then adenine succinase is going to be involved here, right? So again, you will be adding one amino group and following which the adenosine monophosphate is formed. The next way is IMP dehydrogenase. There is the enzyme IMP dehydrogenase that converts inosine monophosphate to xanthosine monophosphate. Yes, that converts to xanthosine monophosphate. Yes, and a transaminase by involvement of glutamine, it forms guanosine monophosphate, which is GMP. So from IMP we get AMP and GMP, right? So those are the two purine nucleotides, final major purine nucleotides that we need. And from INP, AMP is how it is formed. It is formed by addition of one more amino group, and thus adenosine monophosphate is formed. So whereas guanosine monophosphate, first you are going to convert it into xanthosine monophosphate, after which you add glutamine, that is you add the nitrogen group, and nitrogen group will give us guanosine monophosphate. So what is the amino acid involved here? It is going to be the aspartate. Yes, what is the amino acid that is involved here? It is aspartate. So as you can look in the structure, no? Carbon, carboxyl group, amino group and CH2COOH. Okay, now here, whereas in the formation of guanosine, what is the amino acid that is involved? It is glutamine, right? Yes, so this is going to be the synthesis of purine nucleotides, purine mononucleotides. And the important points that you have to remember, which step is the committed step or rate limiting step that I told you? It is the PRPP glutamyl amidotransferase. Okay, and the intracellular and the concentrations of glutamine, of course, will and also PRPP, because the first amino acid that we are going to add is glutamine. Also PRPP is going to be our substrate. So both of which will have effect on the rate of the purine synthesis. And the products will be AMP, GMP, IMP. So if there is increase in products, it is of course going to inhibit the de novo synthesis. And it is also involving utilization of ATP molecules. So now, how is it regulated? So regulation is very important. So from now on, maybe the steps of purine synthesis is not that important for your exams, but from now on, it is going to be very important for your exams. How is purine synthesis regulated? So hepatic purine biosynthesis is going to be stringently regulated. Yes, it is going to be stringently regulated. Okay, now AMP and GMP will feedback regulate PRPP glutamyl transferase, amidotransferase. I'm again repeating it, no? Again and